All right. So we talked about the law of segregation. Now we're going to look at the law of independent assortment. These two laws, law of segregation and law of independent assortment, are very, very similar. So it's important that you notice the little differences in them. And they are different the more you get to know these laws. However, when you're first learning them, it's easy to get them confused. So the law of independent assortment basically says that alleles for different traits are distributed to the sex cell and the offspring independently of one another. So the big thing with the law of independent assortment is we're looking at alleles of one trait and their relationship to alleles of another trait. So we're comparing really, or looking at the relationship between alleles of different traits, whereas the law of independent segregation is only looking at the alleles of one trait. So here, or I should say genes, so here the law of independent assortment, it says alleles for different traits, and different is in red because that's very important that you understand. You're looking at more than one trait. And it describes, underlined, bolded, and in red, two or more genes relative to one another. That's the meat and potatoes, folks. That the law of independent assortment is looking at more than one gene, two or more genes basically, and their relationship to each other. And what it's basically saying is that, now, does this law always come into play? No, but it's saying genes segregate independently. If you have brown eyes, it does not mean that you're going to have brown hair. That is the case. If every brown-haired person had brown eyes, then we would say the law of independent assortment does not come into play. So remember, these two laws, the law of segregation and the law of independent assortment, were created by Mendel when he discovered these pea plant traits. And they follow all his rules. Do all genetics that we study follow the law of segregation, the law of independent assortment? No, not all traits will follow this law of independent assortment because we have some traits we know that are so-called linked, okay? We can show the law of independent assortment by dihybrid crossage, showing how these alleles will segregate independently. Sorry about that. Sorry, my phone is being a little bit naughty. All right, so here we are with this law of independent assortment. And at which stage does the law of independent assortment, at which stage is it illustrated? Well, that we have talked about in our previous unit. It occurs in meiosis 1 during metaphase, okay? And here we are in metaphase. And here we are in meiosis 1. Metaphase 1. The exception would be if genes are close together and they're linked, okay? If genes are close together and they're linked, then we find, suddenly this isn't real, but if you have brown eyes, you're going to have brown hair. That would be the case. Or let's take our corn. The corn example would be if, let's say the purple corn if it's purple color, then that means it's going to be sweet corn. And if it's clear color, it's going to be the starchy corn, okay? So that would be the exception. It would be linked. The law of independent assortment is saying that these genes are not inherited together, okay? So the exception here is if we have linked genes. And they have to be close together because remember what we learned about crossing over. If they're far apart, then they might get separated when they cross over. Now, does a corn, so this is that corn example I was talking about. This is saying so does a corn have to be yellow to taste sweet? No, not according to the law of independent assortment and if it isn't, if the genes are not linked, okay? the color of the corn, and if it tastes starchy, which is like the starchy or the normal kind of tasting corn, or sweet corn, is not linked at all to corn color, corn kernel color. So no, if a corn is yellow, it doesn't mean it's going to have to taste sweet. Now, this is an equation you need to know. You'll need to know this for your worksheet for the second assignment this week. But 2N, and you'll also have to know this for your test, 2N equals the potential gametes. Now, those are the sex cells. N is the number of heterozygotes. So basically, you would take 2 times the number of heterozygotes of the parents. So if these were the parents, remember, we're always got to do 2 traits if we're doing independent assortment. How many potential gametes could be produced? So you would say N is the number of heterozygotes. So in this case, if this was you only look at one parent, this was one parent, and this is the purple, and this is, if it's starchy, you have 2 heterozygotes. So the number would be 2 to the power of 2, which would equal 4. Okay, that's how we could tell the number of possible gametes, different possible gametes that parent could produce. 2 to the power of 2 is 4. Okay, it's a quick, easy way to figure it out. You could also draw it out, but it'll take a while. So the law of segregation, we can show by a monohybrid cross, and it is only a single trait. Again, this is stating that all individuals receive 2 factors, 1 from each parent. Okay, the law of independent assortment. This is where we're looking at 2 or more traits. We're saying, how are traits related to each other, if at all? Usually they're not. Genes on separate chromosomes will assort into gametes independently. Okay? So notice the major differences. This is looking at a single trait. This is looking at 2 or more traits, okay? But one thing they both have in common is that they both occur during metaphase 1. All right, and who's the exception? Linked genes. Okay, and we will talk about a lot of examples. So here's some great examples of test questions. What is the main difference between independent assortment and the law of segregation? Hopefully you've got that by now. Independent assortment is when you're looking at 2 genes or 2 traits and their relationship or lack of relationship with each other, and it can be demonstrated by a dihybrid cross. The law of segregation basically says you get 1 trait from your mom, 1 trait from your dad. It's 1 allele from your mom, 1 allele from your dad. It's looking at a single trait, and it's saying, okay, when you have a sperm cell, it's only going to get 1 copy of the homologous chromosome. Then that copy, if let's say you get that 1 copy, upon fertilization, it will combine with the mother's copy. So you always get 1 allele from each parent. Again, this is just 1 single trait we're looking at here. The individual with a genotype A, big A, little a, big B, little b, big C, big D, little d, can make many kinds of gametes. Which of the following is the major reason? Well, let's look at these answers. The tendency for dominant alleles to segregate together, hopefully that doesn't even tempt you. Recurrent mutations forming new alleles. Well, that's true, but not necessarily all happens. It's not the major reason. Crossing over occurs during prophase 1. Well, that does occur. Different possible assortment of the gamete, of the chromosomes into the gametes. This is what we're really looking for. That is the most correct answer, D. Okay? Now, this individual with that genotype, how many different gametes could they form? Well, this is a great way to work this out. 2 to the power of n, and how many heterozygotes do we have? 1, 2, and 3. So 2 to the power of 3. That would be your equation for the different type of gametes. So 2 times 2 is 4, times 2 is 8, okay? It can get really tedious as these numbers get bigger. All right, we're going to talk about Punnett squares next lecture.